In this lecture, we're going to discuss microbial growth. So when we're talking about microbial growth, we're talking about an increase in numbers of cells of bacteria, not an increase in the size of the cells themselves. We're going to focus on the requirements for growth, whether they are physical or chemical. Physical requirements include things like temperature, pH, osmotic pressure. Chemical requirements are things like carbon, the most important element, or nitrogen, sulfur, and phosphorus are other really important elements. Trace elements, oxygen, which is a very big one, and other organic growth factors. So let's talk about temperature first. Okay, each different species has a limited range of temperature that they grow well at. Okay, so that's their optimum temperature. There's a minimum temperature that they can't grow below and a maximum temperature that they can't grow above. But from that optimum, the drop-offs to minimum and maximum are fairly quick. We can break microbes down into 5 categories based on their range of temperature preference. Okay? First, we have our psychrophiles. These microbes love the cold. They can grow from -10 degrees Celsius up to 15 degrees Celsius. Psychrotrophs grow well between 0 and 20 or 30 degrees Celsius. These are ones that can grow in your refrigerator. They can cause food spoilage. They're more common than psychrophiles. Going up though, mesophiles are our moderate temperature loving bacteria. Most bacteria are mesophiles. This range is anywhere from 10 to 50 degrees Celsius, but the optimum is right around 37, which happens to be the same temperature as us. They grow well at our body temperature. They commonly cause food spoilage. They are the most common type of microbe and the most human pathogens. And then we have our thermophiles, which love the heat. They can make endospores that survive very high temperatures. They grow from between 40 and about 72 degrees Celsius. The highest ones are hyperthermophiles. These are usually our archaea, but some bacteria can be in this category as well. They grow at very, very, very high temperatures, greater than 80 degrees Celsius, oftentimes associated with volcanic activity. So hot springs, deep sea thermal vents, those sorts of things. That's where those bacteria like to grow. Here's an example psychrophile that lives in very cold temperatures. It's Chrysobacterium greenlandiase, named for its being discovered in Greenland. It was found deep inside a glacier. It's hypothesized that it's lived in this glacier for more than 120,000 years. It's able to live for long periods of time in a very extreme environment, very low temperature, very high pressure. They've got the weight of 3,000 meters of ice on top of them. They live in a very low oxygen environment, in a very nutrient poor environment. They're a gram-negative bacillus bacteria, and they actually are orange in color due to production of an orange pigment that they make. So pretty cool. Put my thumb in it. Sorry. All right, so let's talk a little bit about your refrigerator and why it works. Not so much how it works, that's more chemistry and physics, but why it works. So when you bring bacteria down to a temperature that is much far below their optimum temperature, they stop metabolizing, they stop growing, they're not making enzymes, they're not making toxins, they're not multiplying in number, but they're also not dead. This state of bacterial living is called bacteriostasis. It's maintaining one level, okay? It's not growing, it's not metabolizing, but it's still alive, bacteriostasis. So our food, when we put it in the refrigerator, is brought down to a temperature that keeps it at bacteriostasis, which is why if you put your leftovers in the refrigerator for a few days, you know, they're still fine, but if you take them out and leave them on the counter for a day, they're going to be spoiled because that bacteria was still in the food, it just hadn't grown. And when you remove that cold temperature and bring it back to room temperature, that bacteria will again grow and your food will spoil. So our refrigerators work by keeping the microbes in our food at bacteriostasis. All right, another physical requirement for growth is pH. We know that most microbes and most living organisms in general live in the somewhat near neutral range, okay? Most bacteria grow well between about 6.5 and 7.5. So neutral is right at 7. So this is fairly neutral. When we're growing bacteria in the lab, they make acidic waste products. And so that tends to make their environment more acidic. And so often in media that we use in the lab for growing bacteria, they add buffers, which prevent changes in pH for a while. Not forever, which is why you can't grow bacteria in the same culture forever, because eventually the waste products will overwhelm the bacteria. There are some bacteria called acidophiles that grow well in acidic environments. They are rare. Very few live below pH 4. And that's one of the reasons why acidic food generally spoils less. It lasts a little bit longer. However, molds and yeasts grow well between 5 and 6. So this is a little bit lower than bacteria, but not a lot lower than bacteria. So when we grow mold and yeast in the lab, we use a media that's a little bit lower pH because the bacteria don't usually grow on that media, but the mold and yeast and fungi will. All right, here's an example of an extreme acidophile. This is an archaea, Ferroplasma acidophilium. It's found in the drainage from mines, specifically mines that are mining pyrite or fool's gold. It's iron sulfide, so it's made of iron and sulfur. And when it's exposed to air and water, it actually makes chemical reactions that produce sulfuric acid and dissolved iron. So this, as you can imagine, is very bad for an environment. Sulfuric acid is a very, very strong acid. And this acid that's made runs off into the local environment, streams and rivers and those things. And it's a huge contributor to pollution caused by mining. So Ferroplasma acidophilium likes to grow in this super acidic acid mine drainage that's full of sulfur and iron. It normally grows at a pH of about 1.7, which is a little bit more acidic than your stomach acid. Stomach acid is about 2. It doesn't have any cell walls. It has these lipids that give it some stability in a very acidic environment, which causes these really weird irregular shaped cells. And because it lives in such a sulfur and iron rich environment, it actually has proteins inside it that revolve around iron. They need iron. So those are iron centric proteins. And they get their energy from actually breaking down ferrous iron, which is a type of iron. So pretty cool, very unique species of archaea that has adapted to its very extreme environment. So Ferroplasma acidophilium. There's a couple of links down here if you're interested in learning more. Our final physical requirement for growth is osmotic pressure. Now, all microbes are living in some sort of liquid solution, whether it's water or a gut or something. It's always interacting with the environment around it. And so osmotic pressure is really important. So remember, osmotic pressure is the difference in solute and solvent concentrations outside and inside the cells. And for the sake of simplicity, we'll use salt as our solute and water as our solvent, okay? So in an isotonic solution, the salt and water concentrations inside the cell and outside the cell are equal, okay? The cell is happy and fine. In a hypertonic environment, you have a high salt concentration outside the cell. Remember, water follows salt, so water goes outside of the cell to try to dilute that high salt concentration and it comes out of the cytoplasm, so it causes plasmolysis, or the shrinkage of the cytoplasm inside the cell, and this can kill the cell. So this is one reason why high salt or high sugar foods can help prevent microbial growth. So in the days before refrigerators, people used to salt their meat, and that helps preserve it essentially, because bacteria can't grow in such a salty environment. Some bacteria, however, like high salt environments, and some archaea. Those are called halophiles. Halophiles love the salt. And remember, the opposite of hypertonic is hypotonic. That's when you have a very, very low solute concentration around the outside of the cell. You have more salt inside the cell than out. So water goes into the cell because water follows salt. Higher salt inside the cell, water moves in. This can cause the cell to burst or lyse. One of the reasons that bacteria have such a strong cell wall made of peptidoglycan is to help prevent against this bursting. Right, here's a video on osmotic pressure in case you need a refresher. I think I already linked this for you, but I'll link it again. All right, let's talk about chemical requirements. So carbon is our biggest one, right? Everything needs carbon. We need it for structure, proteins, energy, everything. If you are a heterotroph, that means you get your carbon from other organic sources. You usually ingest things that have carbon in them. If you're an autotroph, like a plant or an algae, you get your carbon directly from carbon dioxide in the air, which is why plants consume carbon dioxide and essentially give us oxygen. If you took back a bacterial cell and dried it out, half of the weight of that cell is carbon. Okay, so carbon makes up a very significant proportion of any living cell. Okay, our other important elements are nitrogen, phosphorus, and sulfur. Nitrogen is in amino acids and proteins and nucleic acids, and it's very, very important. Some bacteria can decompose proteins or break down proteins to get nitrogen out of them. A lot of them will use ammonia, which is a nitrogen-rich compound, or nitrate, which is also nitrogen and oxygen mixed together. A few bacteria can actually use the nitrogen in our air. The air we breathe is actually mostly nitrogen. But a lot of living organisms can't use it, except for some special species of organisms, such as the nitrogen-fixing bacteria that live in soil or in plant roots and help take the nitrogen out of the air, and they actually turn it into ammonia or nitrate that plants can use. And so it puts the nitrogen into the food web that way. But for the most part, organisms gain nitrogen by ingesting things. Phosphorus and sulfur, also very important. If you think about all of the things that have phosphorus, our cell membranes have a phospholipid bilayer, DNA, RNA, ATP, phosphate, lots of phosphorus needed in our cells. And then the fourth and least of these four, but still very significant, is sulfur. Sulfur is important in a few of our amino acids and some of our vitamins. And then there are trace elements and other growth factors. Trace elements are other elements that are not part of these four, or oxygen. Things like iron, copper, zinc. We need them, but in very small amounts. And then we have our organic growth factors, vitamins, amino acids, those kinds of things. Now let's talk about oxygen. Oxygen is probably the most important thing for growth, or the lack of it if you're an anaerobe. All right, so if you are an aerobe, you use oxygen. O2 is the oxygen that we breathe. You use O2 to produce energy, right, through cell metabolism. If you are an anaerobe, you don't use oxygen, you use fermentation. Okay, if you're an aerobe, you can be an obligate aerobe, means you absolutely must have oxygen to live. You can be a facultative anaerobe, which means if oxygen is present, you will use it. But if it's not present, you can actually switch your metabolism over to an anaerobic metabolism and ferment stuff instead, but you prefer oxygen. Or you can be a microaerophile, where you need oxygen, but you can only tolerate very low levels of it. Okay, anaerobes don't use oxygen. There are two categories of anaerobic bacteria. We have our aerotolerant anaerobes. They can't use oxygen at all. but they can be around it without being harmed. We have our obligate anaerobes. They don't use oxygen either. The difference here is that they are harmed if they are living in the presence of oxygen. Why? Because they're harmed by these things called superoxide radicals. Superoxide radicals are very dangerous forms of oxygen that can occur in cells when oxygen is being used for metabolism. It's kind of like a byproduct of metabolism, among other things. And it's very harmful to cells, and most cells will actually make enzymes that break down these superoxide radicals. Obligate anaerobes, however, don't make these enzymes. They can't break down those radicals, and so they can't live in the presence of oxygen at all. Here's a great summary slide. We're going to see in lab that we can grow bacteria in a special kind of media called thioglycolate broth. And the bacteria will actually grow in certain locations in the tube depending on what their oxygen requirement is. Near the top of the media, there's oxygen, and as you go further down deep into the tube, there's no more oxygen left because it has special chemicals in it that reduce the amount of oxygen that's present as you go deeper into the tube. So your aerobes will grow on top, your anaerobes will grow on the bottom, and the rest will go in between. There will be more detail on this on the module for lab this week. There's a really great video here explaining reactive oxygen species. I will put this in our module on Canvas. I strongly encourage you to watch this video. The chemistry can be a little bit intense, but don't worry so much about the chemistry. Worry about what reactive oxygen species are, how cells can get rid of them, and how they can actually be medically relevant. This is actually a really, really interesting video that explains some medically relevant oxygen radicals. Okay, here's a summary of that video. Essentially, when you have free radicals, you have oxygen that has unpaired electrons. So it either wants to steal electrons or give them up, okay? And they can make very toxic molecules until that balance of electrons is satisfied. They can make peroxide, superoxide radicals, and singlet oxygen. There are some enzymes that our cells make that get rid of these, like superoxide dismutase and catalase. Catalase is a very important one in bacteria, so we're going to be talking about that one a little bit more as we go through the course. Ozone is another oxide radical, and then the hydroxyl radical, OH, with a little dot next to it. This is probably the most toxic of them all. All right, so 3 enzymes that can help get rid of our oxygen radicals: superoxide dismutase, catalase, and peroxidase. All right, how and where bacteria grow? Well, some of them grow in biofilms. We've seen that already. Remember, a biofilm is an organization of bacteria that grows on the surface of something. It secretes a whole bunch of glycolates, which gives it a slimy layer on top that kind of isolates it from the rest of its environment. So it gives it this nice little happy area of bacteria where they're protected from the environment and they can share nutrients and plasmids and things like that. Okay. We can also grow bacteria in the lab. That one is obviously very relevant. How do they reproduce? How do they grow? Bacteria divide by binary fission, most of them. That just means that they duplicate their genome and split in half. Simple, unchoreographed, no checkpoints, no nothing. Some can reproduce by budding, where they make a little outlet of their cytoplasm and then they just kind of grow a new cell into that and then pinch off. And then there are a couple of other ways that we'll talk about when we get to the fungi a little bit more. But we're going to focus on biofilms and in the lab. So here's what binary fission looks like. Okay, the cell gets longer, the DNA is replicated, the cell wall and plasma membrane begin to kind of pinch it off in the middle. The growth is complete through the middle, and then it breaks into 2 cells. There's a couple of great videos here that kind of animate the process for you. Really great to kind of see it happen. Here's what budding looks like. Yeast and some bacteria will reproduce by budding. They kind of make a little offshoot of their cytoplasm and then kind of bubble out new DNA into that, and then that pinches off. And then some fungi and a couple of species of bacteria can actually reproduce by making different spores and things like that. Like I said, we'll talk about that more later. So let's talk a little bit about conjugation. I know we already talked about this when we talked about genetics, but this is the one way that bacteria can actually transfer genes from one cell to another, and those recombine into a new, into the recipient's genome. So it's called conjugation. The plasmid gets copied through this structure called a pilus from the original bacteria or the donor to the recipient bacteria. And there's a video of it here as well. All right, when we're talking about bacterial reproduction, we're talking about growth of numbers, right? So the time it takes for one cell to divide into two, and then those two cells divide into four, those four cells divide into eight, and that number keeps doubling. So as you can imagine, over time, that number of bacteria goes up very, very quickly, depending on how long that division takes. E. coli, kind of our lab rat of bacteria, if you will, grows very, very quickly. It has a generation time of 20 minutes. So that means one brand new E. coli cell will double itself in 20 minutes. So E. coli grows very quickly for us in the lab, which is one of the reasons we use it so frequently. So we can say that bacterial growth is logarithmic in that the numbers go up very, very, very fast. And it's easier to plot them on a scale that goes up by the tens. Right. There are 4 different phases of bacterial growth, and they each have a unique name. In the lag phase, that's the first one, bacteria are getting ready for growth. They're making enzymes and other macromolecules, and they're just kind of doing their thing. Okay. And then during the log phase, they start dividing by binary fission, and their numbers go up very, very, very quickly. They remain in log phase until they get to stationary phase. In stationary phase, you don't have any new numbers of cells because the numbers of cells dying is the same as the number of new cells. So the total number stays the same. In this stage, you're using up all of the nutrients, you're accumulating waste products, and you're changing the pH in your environment. And once that gets overwhelmed, cells start dying, and you have cell death exceeding the numbers of new cells, and you have a very sharp decline in your population, and that's the death phase. Right, so lag, log, stationary, death are your 4 phases of growth. Here is the same information, different picture. All right, let's talk a little bit about biofilms. So biofilms are when you have a group of microbes attached to a surface, right? So here's a picture of one. They secrete glycolax on top of them that gives them this nice protective layer. They can grow within that glycolax, okay, and it helps them attach to a surface. Some advantages to being in a biofilm are that they can share nutrients, they're sheltered from harmful environmental factors. This also includes being sheltered from the effects of different antibiotics. And it helps facilitate gene transfer. So since there are so many cells so close together, they can share their plasmids more readily. A great example of biofilm that we deal with every day is plaque on our teeth. Okay, this is a picture showing you how much plaque there is in hours post brushing. Okay, so 1 hour, you have a little bit. 2 hours, you have more. 3, 4, it starts to be a lot. 5, it's really a lot. And then they're breaking off from that biofilm and going to other places and making more. So it's a vicious cycle, if you will. But it doesn't take very long for bacteria to grow into a large biofilm. Biofilms are also medically relevant. A lot of infections involve biofilms. It can be bacterial or fungal. They're really common on medical devices like catheters, mechanical heart valves, those kinds of things. They're very hard to clean because of that layer of glycolax. They're 1,000 times more resistant to microbicides like antibiotics or disinfectants if you're trying to clean them off a surface or a piece of equipment. That's one of the reasons some new products have antimicrobial chemicals incorporated into them to try to prevent biofilms, but it doesn't always work. There is a great and optional TED Ed video. It's a TED Talk on how bacteria talk to one another. It's about 20 minutes long. You don't have to watch it, but if you have time, I really encourage you to. The woman who hosts this TED Talk is a really, really great speaker, and she's done some really interesting research on how bacteria communicate with one another. And then there's a video down here on how bacteria form in a biofilm. It's a great animation to watch. All right, let's talk a little bit about how we grow microbes in the lab. So unfortunately, we don't have any hands-on experience with it right now, but let's talk about kind of the theories behind it and the stuff that we use. So anything that we grow bacteria in the lab, we call media. Essentially, any kind of media is either liquid or solid material that is really, really rich in nutrients and prepared for microbial growth. It's going to have nutrients in it, moisture, oxygen or not. It's going to have the correct pH for the bacteria that you're trying to grow. Usually it has buffers in it to prevent that pH from changing. And you're going to also grow your bacteria at a temperature that they like to grow at, whether in the incubator or not. There are kind of two overall kinds of media, solid media and liquid media. The solid media is made of agar. Agar is a substance that's kind of like Jell-O, but a little more firm. And we use it because bacteria, unlike gelatin, bacteria can't digest agar. It's actually made from a kind of algae, red seaweed, and it works really well for us because it solidifies at room temperature. But when we need to sterilize it, because all media is sterilized before bacteria are grown in it, it's liquid at the sterilization temperature, so it makes it easy to work with. If we're using purely liquid media, that's called broth media, and it's the same exact thing. It's got nutrients in it, it's got peptones or proteins and things like that, but it's just liquid form. So really the only difference between liquid media and solid media is that the solid media is liquid plus agar mixed in, okay? We always start sterile so that we know that nothing is already in the media except for the stuff that we're trying to grow in it. The bacteria that we put into the media to grow is called the inoculum, where the act of introducing bacteria into media is called inoculating, okay? And then a culture is just growing bacteria in the lab, so some terms for you there. Okay, media has to give us an energy source and growth factors necessary for growth. There are two overall kinds of media. One is called chemically defined, where you know the exact chemical composition of every single thing in that media. You know how much glucose is there, how much sodium chloride, how much all of these different things are present. And you can match those to what you need for your bacteria. There are fastidious organisms that require very specific growth factors because you're taking them out of their natural environment. So you have to try to recapitulate that natural environment as much as you can in the lab. Otherwise, they may not grow. And actually, a large number of microbes we don't know a lot about because we can't culture them in the lab, or we haven't figured it out yet. The opposite of chemically defined media is complex media. Complex media is much less specific, but it contains the same categories of stuff. It's got extracts of meat or yeast or plants. Extracts are essentially the broken down components, right? So if you think about meat, it's made of protein, right? So broken down meat is going to have a lot of proteins in it. So that's where your proteins come from. Sugars, same thing. Peptones are partially digested proteins, so you get those amino acid building blocks. Okay. A lot of energy and nutrients come from peptones. Vitamins and growth factors come from the meat extract. Okay, most bacteria and fungi can grow fine in complex media. Most of the ones that we grow in the lab, that is. And then, like I said, some microbes can't be grown in artificial media at all. They need conditions that we can't recapitulate in the lab. There are some ways to culture bacteria in the absence of oxygen. Remember, some bacteria can't be in the presence of oxygen at all. So there are ways to make different chambers that have oxygen removed from it, so they're isolated from the air that we breathe. The most basic way to do that is in a jar. It's called a brewer jar. It's a special jar that essentially removes all of the oxygen from the air. On a much larger scale, you can use these big chambers. Bigger picture of the chamber is essentially like a biosafety cabinet that you would use if you needed to work sterilely with things, but it also completely isolates the air and is completely anaerobic inside this environment. There's no oxygen at all. Okay, we're going to skip this slide. There are different kinds of media, and we'll see this in lab in a couple of weeks. There is media that can be, so there's media that everything can grow on, right? Like I was just telling you, complex or chemically defined media. In our lab at River Valley, we use nutrient broth or nutrient agar. It's chocked full of nutrients and bacteria love to grow in it. There are lots of other medias that you can use depending on the results that you want to get. Some media is selective in that it suppresses unwanted microbes. Okay, and it encourages, therefore, the microbes that you're looking for. Some media is differential, meaning that when you grow different species of bacteria on it, different species will be different colors. So it gives you something you can visually use to identify differences between species. And some media is both selective and differential. So here's an example of a media that we would use in our lab at River Valley. It's called mannitol salt agar. It has mannitol in it, which is a sugar alcohol, and it has a high concentration of salt. So it is selective in that it has a high salt concentration. So gram-positive bacteria will grow on this plate, but gram-negatives won't. Okay, so it suppresses our unwanted microbes, our gram-negatives, and it encourages our desired microbes, or our gram-positives. And then it has that mannitol, that sugar alcohol in it, that some bacteria can ferment and some can't. So the bacteria that can grow on this plate, the gram-positives, if they ferment mannitol, they will actually turn the media yellow. If they don't, the media will stay red. So this is an example of selective and differential media. Some media is called enrichment media, where it encourages the growth of a desired microbe that's very specific that you want to get. This is usually used if you are using a fecal sample or an environmental sample where you know that that bacteria is probably there, but it's in very low numbers. So you're going to try to really encourage the growth of that one specific bacteria. Okay, a little bit more terminology. Okay, when you're growing bacteria on solid media, you can see these little circles of bacteria that will grow. And those little circles are essentially clumps of bacteria that start off as one single cell, and they grow and they grow and they grow and they grow until they have a lot of cells in this little ball, and those are called colonies. Okay, when you have a lot of bacteria on a plate, so as I'm moving the mouse from these individual colonies through, see how you have more colonies closer together, and then you can't even see the individual circles anymore because there's so much bacteria present. But when you get into these individual colonies, you have essentially pure cultures of bacteria that arose from one certain kind of cell. Okay, and this method is actually called the streak plate method, where you start with a lot of bacteria and then you dilute it and streak it some more, and then you dilute it and streak it some more until you go from a very dense sample of bacteria to a very, very dilute sample where you have single cells that are spread out, and you can get pure colonies that arise from those one single cells. Let's talk about preserving our bacterial cultures. There are a couple of ways to do it. In the short term, you can throw them right in the refrigerator. They will go into bacteriostasis, just like the bacteria in your food. And this is actually how we store a lot of microbes in the lab that we use frequently. For long-term storage, you can deep freeze and do some cryogenic freezing. at -80°C usually. You can thaw these and reculture them later. You culture them in a special media that has a lot of glycerol in it, so it helps kind of cushion them and protect them from the cold and from being shocked from the cold when they get put into the freezer. Or you can totally freeze dry your bacteria by lyophilization. So that's when you freeze your bacteria and you vacuum out the water and essentially turn your bacteria into powder. And it can remain like that for years and years and years. And you can revive it just by rehydrating it with regular culture media. Okay, and that's the end of our lecture. Thank you.